and we get started. All right, we have some people coming in. Welcome, everyone. We'll give it a few seconds for people to join. And there we go. All right, welcome everyone to Renegade Research Clinicians Roundtable. If this is your first time with us, thank you for joining us. I'm Isabel, CEO of our organization, and we are a patient and caregiver-led decentralized 501c3 nonprofit, focusing on complex multi-system chronic conditions such as ME/CFS and Long COVID. Within this, we have 3 areas of focus: research that's agile and patient-centered, education to increase the number of clinicians treating us, and practical advocacy around barriers to care. Our work is made possible by our wonderful team of volunteers who move the wheels of our organization. Most of us have a personal interest in advancing research and care for these conditions. To stay in touch with us and hear about upcoming events, please subscribe to our newsletter and follow us on social media. We will be posting the links as well as other announcements for you to have handy throughout the presentation. Our work also depends on donations of all sizes. You can help fund our work by donating to our funding campaign and also by sharing our campaign with others who can donate. Today's roundtable is going to be with Dr. Kaufman and Dempsey. They really need no introduction, and to maximize our time today, we will link their bios and previous roundtables as well, as well as the other announcements that I mentioned. And without further ado, we will begin our presentation today. Please know that this is not medical advice. This is for educational purposes only. Thank you, and I'll give it to you guys, Dr. Kaufman and Dr. Dempsey.
Okay. Well, where do
we start?
Where do we start?
Well, let's start with, what you have been observing with using GLP-1, GIP receptor agonists, what you're seeing in your patient population, how you're using it, and what are the results that you're seeing, and what led you to begin using this medication, as well as what that is happening in clinical practice and the upcoming research.
Sure. All right, I'll jump in first.
Sure.
Partly because it's relatively fresh in my mind since I did a talk with RECOVER on this. So I was trying to think of the history of how we got into this, especially with Tania, and this is through this online group that we have of over 800 physicians and other types of clinicians. And we're able to search the archive, and I did this in preparation for this talk, and there was an email in 2022 from Larry Afrin saying, gee, the most amazing thing just happened. I have a patient with terrible mast cell activation syndrome who's trying to lose weight, and her primary care doc put her on, actually, I think it was semaglutide, right, Tania? I don't think it was.
I think back then it was.
Yeah, it was
right.
I also just learned it's semaglutide.
Semaglutide, yeah.
Okay, semaglutide. And her mast cell symptoms just disappeared. And it was one of those Larry Afrin, you won't believe these emails. I mean, you won't believe this happened type of emails. And it just exploded from there on Masterminds, on this listserv that we're on. And as often happens for me, I started trying it right away, and I had the same experience. And I think it sort of took off from there. We quickly migrated over to tirzepatide, and we can certainly talk about why we think that's better. And I think the key piece is that by using low doses, most of our patients, we don't want them to lose weight, or they don't want to lose weight. And the low doses work for what we wanted without the side effects of the weight loss doses. So it was a very kind of interesting serendipitous moment in medicine, as often happens when new drugs or new uses are discovered.
Yeah. So I had a similar experience to Larry Afrin. I had a patient back in probably around that same time, and that's why there was so much excitement. I had this patient who has a history of a traumatic brain injury after MVA. So she's now, let's say, in her late 40s, early 50s, but in her 20s, she had a major MVA motor vehicle accident with a traumatic brain injury that she has done quite well with. I mean, she's a brilliant woman who has achieved a lot, but she struggles a lot with word finding, some brain fog. And she also has, of course, MCAS and a history of vector-borne infections, Lyme, Babesia, Bartonella. And I've been treating her. She was one of my patients from my original primary care practice before I went on my own. So I've probably known her now maybe close to 20 years. And so, she's struggled with her weight for a very, very long time, and has some like very strange appetite issues. Like she can go for days without eating, and she'll never lose weight. She'll gain weight. She just doesn't often feel like eating, and when she wants to eat, she can. She has some nausea. A lot of it, I think, related to her MCAS and her infections. But she did quite well after I treated her infections. I actually used a Dapsone protocol back probably in 2018 maybe. And then so in 22, we were talking about her weight, which wasn't budging. She was getting really frustrated. And we sort of said, well, listen, she met the criteria for the FDA for approval for the GLP-1. And at that time, I think it was 22, 23, uh, I think Manjaro had just come on the market. I can't remember actually. I have to look back at her chart if we started with the semaglutide or we started with tirzepatide. But she's currently on Manjaro. But what was so interesting about it is this is before I understood there could be microdosing or anything else. We just put her on the dosage. She had a tremendous amount of nausea, tremendous side effects, but she really wanted to push through, which she did. And she stayed on the 2.5 for some time. But what she said was like the thing that really sparked my interest. She said within a dosage, within 2 hours or 3 hours, the brain fog just disappeared. The words started coming and she could focus and be at work. And then it got to the point where she was, really like counting the days till her next dosage, because towards the end she would notice a little bit of a decrease in that ability, and then she'd take the next dosage and she would feel that way. And so we started brainstorming. I started brainstorming, and I was thinking, what is this, drug doing? Now, the other interesting thing about it was that she's actually lost almost no weight, maybe 5 pounds, but she feels less inflamed. She feels better. We've gotten her on 5 milligrams. That's all she can really tolerate. She doesn't even care, that the weight hasn't come down dramatically because she just feels so much better. Everything looks better. And if you look at her, you would think she has lost a tremendous amount of weight because she does look less of that, has had that edema and puffiness that a lot of our patients have. Um, but she has the brain fog lifted, and her appetite has shifted so that even though these drugs are theoretically causing decreased appetite, what she finds is that it's not, she does have a decreased appetite, but it's a different type of appetite. So before I could barely get her to eat anything for days. Now she actually can feel a little bit of that hunger, and she has a little bit better, I guess, a circadian rhythm with eating that she didn't have before, which I've heard over and over again with a lot of patients, that they used to eat either all day long because they were hypoglycemic, or they used to just skip meals and not really have a plan. On the drug, they feel more regimented. They have an ability to understand maybe hunger signals better, and that's what she has found. So anyway, that was like, my mind was blown. And then after that, and then Larry sharing his experience, and then others sharing their experience, I was like, okay, I'm onto something here. I had a number of other patients who were using it for, again, either diabetes or obesity. I said, well, we've got to find a way to help our patients with MCAS, complex chronic diseases, ME/CFS, et cetera. So that was my story. A little long, but I had to share it.
Yeah, I think it's I certainly in the beginning when I started using it, and I think all of us feel this way, it was pretty mind-boggling to see the response and the fact that it was global, it was systemic. and pretty quick. I mean, I was just starting to search for a quote, because when I talk to patients, I'll often put in quotes what they say to me in my note taking. And I had a patient recently who also, as Tanya's saying, after the first dose, described her decrease in brain fog and all symptoms improving. And then I just got another email from her, or portal message. This is a 21-year-old woman off to college, pretty treatment-resistant POTS, struggles to do the college work, takes her 2-3 times longer than it should, is relatively isolated because of her POTS and everything. And she sends me this email saying, basically, my life has completely changed. Um, I can do my work in 1/3 the time it used to take. I have no more brain fog. And this is, by the way, on 0.5, 0.5. And then she did, I had before and after Nasoline tests, stand tests, and you could even see the difference in her POTS numbers without changing any other meds except the GLP-1. This was tirzepatide. So it's a work in progress, but it is moving really fast.
Yeah. Yeah. It's definitely exciting. I think it's important to note that this is a group of drugs that has some um, research to support its use as an anti-inflammatory. It's being looked at for neuroinflammation, right? So I think it's lots of stuff that we could potentially use these drugs for. But on the same token, there are patients who will not tolerate it, and I want to be clear. We're making it sound miraculous,
Right.
but it's not like anything else we use in our sensitive patient population. It either may not work at all. I've certainly have those patients that really don't notice any positive result, but I also have patients who do very, very poorly and pretty quickly, and it's not it's not a lot, but it's enough, to say that, look, I think everyone has to sort of go down this path cautiously, ideally with a provider who really does understand the nuances of this drug. Because I think what's happening is that there are a lot of doctors out there who are trying to help these patients. I think there are a lot of doctors who don't understand at all what they're supposed to do. I get messaged all day long on Instagram about, what should I tell my doctor? What dosage? My doctor wants to start me at 2.5. Like obviously I can't give advice, but some doctors don't understand the nuances. And so it's not that you shouldn't be treated by them, but ideally those doctors should be reaching out to people who have experience, who can help them figure out, the dosing for the individual person. I just think it's important to bring that up.
Are you seeing any similarities in the patients that are responding, whether that is in the other therapies that they're on or their severity level, or does it seem like it is, random across the board?
It's a good question. I'm not sure I can answer it. Most of the patients I've been using it are long COVID patients, and that may their responsiveness may mean something. May be significant in that that's a shorter course of illness than an ME/CFS with 30 years of history, and a
Yeah.
very well-defined infectious cause for their illness. And I think it's a point worth talking about, because it's come up in some other
Yeah.
trials that the post-infectious fatiguing illness, there's a bunch of different names now. If you have a clear infection cause and start date, those patients seem to be more responsive. That's one of the things we've noticed and learned in the rapamycin trial we're doing. The most consistently responsive patients, meaning they improve on rapamycin, are those that have a clearly identified infectious onset cause. So it may be that, but that's a perfect example of something we need to study. We need to have, a, a group of ME/CFS and a group of long COVID and a placebo group, and see what happens. I want to actually follow up on something you just said, Tanya, about other doctors. I agree with you totally, but I also think that there's not enough of us, of Dr. Malcolm and Dr. Dempsey and myself to take care of all of the people that are desperately in need of treatment. I mean, there's 20 million Americans with long COVID, and that's just Americans. So yes, we don't want other doctors doing that, but yes, I feel it's almost our obligation to try to teach those doctors, because this is not rocket science, and we can educate them if they're interested in learning, and that
Yeah.
would be fantastic.
Yeah. One of the projects in my free time that I'm doing is creating a course for medical providers, practitioners, hopefully for like after the new year, that will teach this stuff. Like, I really think that, we're talking
Exactly.
very generally here and whatever, but we just need to go in and just say, here, this is what you need to do, this is what you need to think about. So I'm hoping to launch something like that in the next few months.
Yeah, that would be great. Sign me up.
Okay.
I think
Can you talk a little? Go ahead.
I just wanted to make some few comments on Dr. Malcolm. Just a few things. So I think the story that Dr. Dempsey and Dr. Kaufman gave of like listening to Dr. Afrin saying, "Hey, this is working." Um, this is why I'm here, for posterity's sakes of the way that doctors, other clinicians are, is we get a little scared when we don't have familiarity with something. And long COVID has been extremely frustrating as a clinician in this space because there are a lot of medicines that are not really that risky. It's not hard to give antihistamines to patients, but there is this wall that a lot of people won't cross. And because it's not in the book. So I really kind of wanted to say, like, I like these talks because there just needs to be a moment in time where we are talking about this. And we have these clinician groups where we get to talk about medicine and like what's working. And we'll go forward and we'll try it on our patients. I was one of those doctors listening to Dr. Kaufman and Dr. Dempsey about this medication working. I wanted to give another example of a patient, long-term chronically ill patient, who used Zepbound, and she noticed that her intracranial pressure symptoms went away. And the way that I practice medicine is I kind of I like to provide as much evidence to my patients. So, for example, Dr. Kaufman, thank you for doing that talk with the RECOVER trial, because I can flash it up on the screen and say, hey, this is what Dr. Kaufman is seeing. It's representing what I'm seeing, like an improvement in patients, and here's some evidence of what's going on. But once that patient told me that, I went to Google Scholar and I found a paper from 2 years ago that showed that exenatide, not sure if I'm saying that one right, one of the kind of older
Mhm.
GLP-1s,
Yeah.
was reducing CSF pressure in the brain. It was a
Yeah.
paper out of the University of Birmingham
I
two
saw
years ago.
that. Yeah.
So
Yeah.
it's just there is data out there. There is stuff. Some of these drugs are very safe. Again, I remember the first time giving statins for long COVID and lecturing people, hey, how many millions of people, how many millions of Americans are on this drug? Again, statins have issues. But there needs to be more of clinicians who are I don't want to say the word hiding behind evidence-based medicine. um, in medical school, we're taught like what is the history of evidence-based medicine? We want to produce evidence, but evidence also just lives in front of us. What's working in front of our eyes? Hey, there's some other doc and PPA who's watching this. This is evidence. There's evidence there that these are working. But you just need to be comfortable with the medications that you're giving because GLP-1s also have adverse side effects.
Yeah, I think you're bringing up a
Yeah.
really important point, Stuart. So, yes, evidence-based medicine is really critical and has been a foundation for the advancement of medicine and science. On the other hand, um,
Yep.
evidence-based medicine is generated by trial and error and serendipity and observation and curiosity and a tolerance for uncertainty. And those are such crucial factors to the whole kind of enterprise. And my concern is that in a lot of the rest of the American healthcare system, because most physicians are employed by corporate enterprises, i.e. the hospital, they're simply not allowed to think the way with the three of us are talking right now. Um, and so it's not just a failure of on our end to try to educate other physicians, it's that they're almost not allowed to listen to the education. And
Mm.
that's a roadblock that I don't know how to get past that roadblock. I mean, I'm sure all three of us, you'll have a PCP that says, I don't know what LDN is. I can't use that. And you just have to hit 3 keys on your keyboard and you'll have a million descriptions of LDN.
Mm.
And now Google AI will tell you in the first millisecond and you're done. But they're either not allowed to because they work in a hospital that says no off-label use, no non-evidence-based use, or they're afraid, thinking, well, I didn't get taught that, so I can't try it. And I don't know how to get past that. And certainly the GLP-1s that we're talking about in this experience is another great example of that. And that's a bigger example because it involves not just a willingness to try something off-label,
Right.
but how does that doctor obtain it? I guarantee you, if he or she works at Mount Sinai, they're not allowed to prescribe a compounded medication. So there's just these roadblocks to care. I'm a little off topic, so I'm sorry, but you ignited me, Stuart.
Yeah, but this is important to mention, and the fact that the FDA did go after these compounding pharmacies and still going after compounding pharmacies, and the pharmaceutical companies going after the compounding pharmacies because of that competition, right, also is troublesome because there's this constant fear now in this community. People are feeling better, they're microdosing. Now they're worried they're not going to be able to get it. And then, yeah, all these other doctors at these institutions who can't get it for them, and that there are not enough of us to be able to prescribe it, right? So it's a little bit of yeah, I don't know, like a crazy situation actually.
Yeah.
So,
One of the things that
but
I hear often from clinicians is the insurance coverage obstacle,
right.
not even with compounding, but with standard dosing. How do you guys get around that issue? Are there any tips that you can give clinicians in the audience for, how to overcome some of the insurance obstacles?
Um, I mean, it's a huge, excuse me, a huge obstacle because it's sort of a double-edged sword obstacle. If the only way you can get insurance coverage is if the physician is prescribing it for an FDA indication that meets the criteria, and then so that's the first piece, which immediately means you can't microdose. I mean,
Mm.
you cannot obtain it
Right.
at a low dose and still get coverage. You can't write a prescription to CVS for Wegovy and say 0.25 milligrams start dose. They'll look at you like you're crazy, and even if insurance was willing to, you just can't get it. So there's
Yeah.
that piece, and then there's the difficulty with insurance in general. So honestly, in my practice, it's almost always compounded medicines and the patient has to pay cash. I will say the cash cost is vastly lower than the commercial cost. I mean, we're not talking
Oh,
thousands of
right.
dollars where patients are having
Right.
to pay literally $1,000 copay or something. We're talking, much, much, much less. The programs now with Lilly Direct, and I think Novo has a uh, direct to consumer site now as well. Even there, my understanding is that Lilly requires that the physician say, give an FDA approved diagnosis, so they won't let the patient buy it with cash.
Right. Right. No, that's true.
And while I'll cut a
So
lot
it's a
of corners, I'm
challenge.
not going to lie about the diagnosis to Lilly. So
Right. Right. So the reality is, there are specific diagnoses that you can use for Ozempic, for Wegovy, for Manjaro, for Zepbound. I have a slide that I presented at and ILADs on this very talk. And so, really, they've expanded it a bit. It used to be very sort of black and white. Ozempic, Mounjaro for diabetes, Wegovy, Zepbound for obesity. Now it's, for the obesity one, right? They'll say you could be overweight with a BMI of 28, I think, and have comorbidities like high blood pressure and other risk factors. The same with the diabetes one. They've extended it with people who have, more comorbidities, right? They just keep expanding it that way. But they still, I mean, many of our patients are not going to fit in those boxes. So there's no way you can get insurance coverage, right? If you can't
Mhm.
fit in a box.
Yeah.
Plus,
So,
even if you can, pulling off the microdosing is very difficult because of the way the commercial products are dispensed. I mean, it's a hard and compounded drug as
Yeah.
well, but at least there we have some flexibility. But that's another difficult piece.
Yeah. Yeah.
so can you help clinicians understand how you make the decision to prescribe this medication and the decision to where to start in the dose?
You want to
Should
go?
I start? Yeah, I
Yeah,
can
go ahead.
start. So look, I think that back in 22, 23, I thought of these drugs more for, again, the standard FDA approved usages. And then for these other patients, right, that didn't meet those criteria, as maybe second, third, fourth, fifth line treatment. I would make sure that I've tried and exhausted all the MCAS targeted therapies. I've, worked on, let's say, their mold issues, their Lyme issues, and then they're still not getting better. Oh, okay, you know what? There's this really great option. Let's do GLP-1. That was where I started. Um, but I have to say, and I think David's shaking his head because I know he's thinking the same thing as me. Now it's becoming first line, which is really interesting, right? So I may not, sure, I may put them on an H1, H2 blocker and then sort of start working on this, but I'm no longer thinking of it as further down the list of interventions. It's higher on the list. So now in terms of dosing, again, especially if patients are normal weight, or especially if they're underweight, right? You have to be very, very cautious on the dosing. The the dosing for patients who, let's say, are a little bit more overweight, but they don't meet the criteria, I still am very cautious. These are my sensitive patients. All my patients are sensitive, so I'm never going to start them at the full commercial dosage. But I think there's no harm in starting as low as possible, maybe a tenth of the normal dosage or less. And again, there's no urgency. Even at that low dosage, people see a response. Sometimes they don't, and then you can increase it. Sometimes patients will tell you that they noticed something with the first dosage. They didn't notice much on the second dosage. They noticed that as they were waiting for the second dosage, the effects were wearing off. So you start to play around with, okay, we increase a little bit, and then, but maybe we also split the dosage. So we half that dosage and they take it twice a week. I have found that to be actually particularly useful for a variety of patients, splitting the dosage and having them so it's less at one time, but then they have more coverage over the course of the week. So again, these are tricks, very individualized. You have to listen to the patient to really know.
Yeah, that's
What to
been
do.
my experience. That's why I was shaking my head or nodding my head, whatever it is.
Yeah.
So, in
That's what I meant,
medicine,
nodding. Yeah. Yeah.
we're generally taught as physicians, you escalate your care gradually and you step in one step at a time, one toe at a time. You start with the least invasive, the least risky, the most evidence-based treatments. And so in the beginning with this drug, uh, in my practice with a patient, maybe I'd start thinking about it at visit 4, 5, or 6 when they're not getting better after I've tried all the basics, the H1s, the H2s, the stabilizer. Okay. Absolutely now, by the second visit, I am considering starting this drug because number one, it works. It's very effective. Number two, in the very low doses, I am not seeing significant side effects. And number three, almost painfully, I say this, why make the patient wait? Why should I wait until the fifth or sixth visit and six months of time pass if I have a drug that may really, be the game changer for them? I just also want to add one other thing on the dosing concerns and side effects. It's not just that we don't want our patients to lose weight. Almost all of them, I would actually say all of them, have autonomic nervous system dysfunction. That is a cardinal
Yeah.
finding in long COVID especially, and therefore they all have, or most have, motility dysfunction in their gut. And so the biggest side effect I'm actually concerned about with the GLP-1, both groups, is making that motility worse. I'm less concerned about weight loss if I compare those two issues. And again, in the low dose, I am not seeing that happen, which is really crucial. We're getting benefit without further injuring their motility problem.
Great. So, if you have a patient that you start on this medication and they're not responding as you'd like them to, where do you go next? Do you go try to maximize the dosage or the frequency, or do you go for the different medications that are available?
So for me, that's the murkiness, the muddiness of trial and error medicine, which frankly is all I do every day. Okay, especially since everything's off label, especially since we don't really understand so much of what we're dealing with. So okay, so the trial is a very low dose of tirzepatide. Patient says, oh, I don't feel a thing. Is that because I chose the wrong drug, or maybe they need a higher dose? So my practice is I will gradually inch that dose up, either until it causes side effects and no benefit, or until I start seeing benefit. So for me, there's no easy answer to what you're asking. I actually I've not switched from tirzepatide to semaglutide in this process at all, partly because I think I've always had success with tirzepatide. I don't know, Tania, have you been switching back and forth? I think you told about one case at ILEDS where you switched.
One case, yeah, one or
Yeah.
two. I had one patient who had um, increasing reactions at the injection site to tirzepatide. So we tried the semaglutide and did better. And then somebody else who felt better on the semaglutide, less inflammation than tirzepatide. Generally, I think tirzepatide is more anti-inflammatory, but again, everything is individualized and everyone is different. So I think there are a couple of points that I agree with David completely. I think it should be mentioned that in people who are getting higher dosages of these drugs, there are reports of the antibody production to the compound and causing it to be less effective. And so they're seeing that, at the, again, commercially available doses. Let's take, for instance, Mounjaro. People are taking, let's say, 7.5, 10 milligrams, higher doses. Over time, the development of antibodies is making it not effective. It's not able to, so the antibodies are binding to the compound, not allowing it to bind to the receptors properly, and the patients are not getting a response. And so some doctors are pushing the dosages even higher to try to overcome the antibody issue. I'm not sure. So I do think, though, we're starting to see this even in patients who are having lower dosages, right? So the point is we don't know how to handle these patients yet. I do think that there are some patients that I've been able to keep pushing a little bit. They're still on a low enough dosage that I feel comfortable. They were stable for a period of time, 3 months, 4 months, maybe longer. And now they say, I don't notice it as well. Okay, let's bump it up a little bit. As long as they keep getting a response, I'm okay with doing that. But I do think this is something that we're going to have to face, because I think sooner or later, the longer we have these patients on it, it's going to be a greater issue. And I don't know how to handle it. I think in some people, let's just take a different community that I know uses a lot of this, and this will be the longevity community / bodybuilding community, where they're using it for shredding or they're using it for longevity. Some of those people are pulsing it. They understand the antibody issue, so they're taking it for a period of time, 12 weeks, and then they're stopping it for a period of time. Then they're coming back, and most of them notice they do feel better on it, so they want to go back pretty quickly. I don't know if that's a good strategy for our patients, and I'm really concerned about doing that. But I bring it up because there are people talking about this. I've talked to a number
Yeah.
of colleagues in the longevity world who are using that strategy. So I don't know yet. I think we need more research. I spoke to a lab director at iLabs who internally did a study on antibodies. He didn't publish it, but he had a number of family members on it. And so he was curious because he had started reading the literature on it. And he said that all of them, I think it was like 4 or 5 people in the family, all of them had antibodies actually already to the drug after a period of time. So I don't think it's really rare. I don't know if it's a problem in every patient. I don't know what we're going to do. I don't know if it means that as the newer drugs come out, we have to change the drug. In 2026, retatrutide will be on the market. We can already compound it, but it's a triple incretin with GLP-1 receptor agonist, glucagon receptor agonist, and GIP receptor agonist. So the question is, would adding a third incretin on there, would that be more helpful for these patients? And would it overcome this issue? But I think it's just, important to just put it out there because I have a feeling we're going to see more and more of it.
Yeah. Yeah. The making antibodies to it, and I'm glad you brought it up. It's certainly an interesting problem and aspect to it, and we see it in lots of other drugs. I mean, in the biologics world, there's a drug called Humira used for inflammatory bowel disease, and it can be remarkably helpful and successful, and then all of a sudden it starts to not work, and it is
Right.
because of antibodies. And you can actually, as a physician, measure the antibodies to that molecule to try to anticipate failure, I guess, or adjust the dose. But constantly adjusting the dose isn't going to solve the problem ultimately if antibodies are developed. And in that world, in the inflammatory bowel world, you change biologic. So your point is well taken that maybe that's an example where if tirzepatide is not working so well after 6 months of use, you switch to semaglutide, which is a different molecule the antibodies haven't developed yet, and maybe you go then back to tirzepatide. These are all works in progress. We got to learn this,
Right.
and we're only going to learn it. Honestly, we're only going to learn it, I think, from people like us that are prescribing it to patients and observing it and taking notes and learning,
And
because it's
trial and
very
error.
hard to get trials to do this stuff.
Right. But I think that because we can't get trials, but I'm hoping there are trials. I've already heard some whispers of some interesting research that may be happening. There's some companies who are really interested in using biomarkers and trying to figure out, I think particularly in the longevity realm. But in the meantime, we published our 47 case series. I think there needs to be, 150 case series with additional observations like in a year. And then maybe it's a 500 case series and we bring in more people, right? David, I think you didn't
Yeah,
get a chance to publish with us, but we gotta get you
yeah,
into
I
this
agree. I
publication,
agree.
Yeah. And Stuart, of course, who's now also getting experience with it. So I think the more and more we can talk about our experiences, the more I hope that there will be interest to do, randomized controlled trials or whatever needs to be done to convince people that this is, useful therapy.
Yeah, I want to just comment on that because I think if I were like a little old resident trying to like look to like guidelines on this, like Dr. Kaufman, you are giving this data to the RECOVER trial, the $1 billion money from the US government for long COVID. And Dr. Dempsey, you are on this paper that was published this month. So this is the data that's out there, and I'm hearing the two experts say, hey, we know something. We know that we're trying, we're seeing things, but we need some help, and we need help by getting more people to do this. So I think the new administration might be trying to collect data like this. Again, I always say long COVID lives here. It's 200 million people. We kick politics out. I don't care about politics. We just need to solve the problem. So I
Yeah.
think that, to always, quote Larry Afrin, it's publish, publish, publish. But we need that, but we just do need more folks kind of seeing, because this conversation about the autoantibodies and lower doses and all this sort of stuff is just new, but you two are the ones that are putting the data out there. So it's just, we got to keep going.
What are you guys doing in terms
100%.
of the other interventions, especially for not new patients that are coming to you, but the patients that maybe you've had for a long time and are now having a good response on this intervention? What's happening with the rest of their profile in terms of everything else that they've been on?
That brings up trial and error again.
Yeah.
Yeah, I mean, if I have a patient, excuse me, when I have a patient who is responding, let's just use MCAS, mast cell activation syndrome,
Sure.
and they're already on H1 blocker, H2 blocker, cromolyn, and ketotifen. So 2 mast cell stabilizers and mediator blockers, and they still have reactions and food restrictions, foods that trigger reactions. And now they're on tirzepatide, and all of that is much better. Then I will cautiously and with anxiety gradually pull off some of those meds and see if they're okay. There's no way I can know in advance without literally going through that process. So it's always anxiety provoking for both myself and the patient. But that's kind of the only way. Same argument for POTS. Many of my patients are on three or more drugs to try to manage their POTS, along with endless fluids and electrolytes. Gradually try to pull back on some of that if possible and see. There's no way to predict. It's only you got to try.
And what are you seeing so far when you do pull back on those medications?
So I have not done a lot of it, meaning I have not yet pursued that with many of the patients, but with some of them, it's so far been successful, meaning their improvements are maintained on the tirzepatide as I pulled off those medications. I have not done that yet with POTS, and this patient that I just described a minute ago, the college student, really opened my eyes to that, because it was unusual to be able to get a pre and post lean tests in such a timely fashion and see the difference, and that difference correlated with her improvement in all parameters. So yes, that would be my next step for a patient like that.
And I have to just add that at least for my patients, I don't have that many patients that I've actually been able to pull off other things yet. I mean, we've tried, and it seems like the patients know what each intervention did. So as we pull away, they notice that they don't necessarily feel as good. So I think that the GLP-1 receptor agonists are obviously making a difference, but I think it's not going to be enough for these patients, at least my patient population. And there's some differences, of course, between our populations, but we're trying, but I've not seen that yet. And so that speaks to, for me, and I know David will agree with this. There's something else there, root cause, things that are underlying that have to be addressed. So if we think about, I hope this is not too controversial, but if we think about the long COVID world, I believe that there's a pretty sizable subset who have persistent infection. And so I'm sure, there's the spike protein thing that people are talking about, and the post-vaccine thing, but I think persistent infection in general, whether it's COVID or whether it's vector-borne or Epstein-Barr or whatever we're talking about, is going to keep the immune system activated inappropriately. And so these are all great tools, and sometimes these are the only tools the patients need. I have certainly seen that. But I also think that for many patients, we do have to continue thinking about, okay, we've stabilized them to some extent. Now, maybe some of the interventions for the persistent infections can be undertaken, right? Otherwise, if they're so sensitive, we can't even go there. So I think it's important to bring
Yeah,
that point
yeah,
up.
yeah, Tanya, very, very good points. I want to comment on it in a couple of different aspects of
Okay.
it. Okay. So
Yeah.
yes, you and I, we've talked about this many times. I 1000% am convinced that COVID and long COVID, a major aspect of it is the reactivation of previous latent infections in our patients. So all of mainstream medicine now accepts that EBV reactivates in roughly 70 to 80% of COVID patients who get COVID. So that's a given. Everybody accepts it. There's lots of evidence. I'm 1000% convinced, Tanya, I believe, is as well, that the same argument, the same finding occurs with vector-borne infections, in
Mhm.
particular Lyme, Borrelia, Bartonella, and Babesia.
Mhm.
That's a whole new concept, in no way accepted by mainstream medicine or infectious IDSA docs. The idea being that people get infected as earlier in their life before COVID, when they were hiking and camping or got scratched by their cat or whatever. They got infected, their immune system controlled that infection, just like when you had EBV at 3 years old in the sandbox and you didn't get sick. And then something comes along to disturb your immune system and it reactivates. So yes, we think about that, and I agree we need to look for it. The 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 thing I want to say about the GLP-1 and what's happening is two things. By reducing inflammation systemically, it is dealing, I think, with one of the foundation or root causes of illness in long COVID and ME/CFS. And by reducing inflammation systemically, it allows that person's immune system to begin to recover, so that we may not even need to treat those reactivated infections. We may have an immune system that now is recovered enough to push it back into latency. I have no proof of that. That's kind of a dream or a goal, but I think it makes a fair amount of sense when you think about these illnesses. To put it another way, to me, the use of the GLP-1s to reduce inflammation everywhere is dealing with the root causes of the illness in a similar way that rapamycin is doing that, by increasing autophagy and decreasing systemic inflammation. And there we have proof, clinical trial proof in human beings, not just mice, that by doing that, their immune system improves and becomes more competent and they get less infections. And that's a crucial piece of information. And I think that's the same direction here. Nevertheless, Tanya, I agree. Yes, we still need to think about infection, and maybe that's the moment to go after it, because as you know, and I
Mm.
know, trying to give a long COVID patient with MCAS and POTS and motility disorder, 3 antibiotics to treat these infections, you can't do it. You have to get
Yeah.
control of all those other things first. So the tirzepatide, these drugs may allow us to get that control so that we can more safely and easily treat the infection if we still need to.
Yeah. Yeah. 100%. Agreed.
Yeah, I 100% agree. And other things that I'm kind of stewing on is, so I'm a part of the millennial generation, the greatest generation alive. And my cohort did a lot of traveling. I have a lot of patients who spent a semester abroad in India, who went backpacking in Africa, whatever. We have no idea what some of these infections might be. I think that there's obviously David Petrino did announce this in public about the data regarding Babesia and Bartonella in long COVID patients and how common this is. But I 100% agree with you, Dr. Kaufman, in terms of, will we get control? I think interesting studies that I would love to do, but this stuff is expensive for our patients, is like, what do the antibody tests show before, directed against these, tick-borne infections before Zepbound or whatever, and then after? Could we start getting signatures of like, oh, the immune system's waking up? There are going to be naysayers, of course, but that would be something that would be really interesting. But I guess I also worry, like, what I agree with you that I think if we get some control, can we finally of the immune system, can we finally then attack the causes of this sort of stuff? And who knows what those things are going to be, because we don't know what, decades of antibiotics use have done to the microbiome. We don't know what extensive travel around the entire world has done. There are a lot of probably things hidden out there that we don't even know about. So, yeah,
Stuart, we may not even need to treat them. That's
we
the
might.
point. I mean, I
Yeah,
don't know, but
yeah,
Yeah,
as
I
somebody
agree with
who's
that.
been trying to treat these infections for 30 years, the thought of not having to treat them, that's like
we've
a miracle to me. I would
Yeah,
would
love
be
it.
wonderful.
yeah,
Yeah.
Yeah,
yeah,
I'm hoping, because if I don't know what it is,
yeah,
I don't want to worsen the patient by giving something that might
right,
hurt them as
right.
well. So let's hope.
yeah.
And we'll have to see.
Well, to that point, I mean, I talk about that a lot, and spoke on that at iLives too. The reality is that these are these are the how do I put this? I think lots of people out there are walking around with quote unquote persistent infections, whether it be, they had mono and so they have some persistence of Epstein-Barr, whether they, live in New York and have had multiple tick bites, or anywhere in the country really, or the world. um, you know, but if I pull in 50 people off the street, right? This is an endemic area where I live, and I pull in 50 people, I assume that many of them will test positive, even molecularly positive via fish test. I don't know the numbers. I wish we could do the study, but 80%, 90%, I think it's going to be fairly high. But a lot of people are not sick. They don't have chronic illness. They don't have MCAS. They don't have a variety of things. And so I do think that there's something about the people who are sick and what these infections are doing and what their immune system is not doing. And so if we get their immune system online and we reduce inflammation, maybe it doesn't matter about these infections, right? I think that's the point that
That's what
David's
I'm trying to
making.
say.
Like
Yeah.
yeah, exactly. Yeah.
I mean, it is a testament to how extraordinary our immune systems and how has
Yeah.
evolved is, because just think about the numbers for a second. So I forget how many Americans have had COVID now. I think, Stuart, I think you said 200 million. I think that's actually correct. It's in
So
that
Dr.
realm, right?
Yeah, Dr. Z has kind of like a theory, kind of like crunching the economic data. He's the head of Long COVID at the VA hospital in St. Louis. So he has that data. But even if it's a tenth of that, it's
Okay,
still a
so
lot.
let's say 200 million, and let's
Mm.
accept the finding that 70% of patients with acute COVID have reactivation of EBV. Okay, so that means 100, is it 140 million? Yeah, 140 million Americans have had EBV, and they were not sick at all from EBV because they
Right.
had a potent, competent immune system. And then they get COVID and their immune system is disturbed. It's disabled or declined. Their EBV reactivates, but out of that 140 million, only in quotes, 10 or 20% of those go on to long COVID and continue to have EBV reactivation, meaning that incredible immune system is then, recovers and knocks that bug back into latency. And that's the argument to some, that we're sort of making, that we need to change our focus to the immune system to try to solve these problems systemically, rather than an antibiotic here and an antiviral there, one step at a time. Now, I'm not saying we shouldn't use those drugs. I'm just saying if we could fix the boss, that would work a lot better.
So a two-part follow-up question to that. One is, are you utilizing this medication with the more severe patients, the mostly bedbound or housebound patients? And regardless of the severity spectrum, is there any patient that you would say, no, I am not going this route for whatever reason?
So mm,
So I'm going to answer the first part first in
yeah.
terms of severity. So I would have answered, if you asked me this question 8 or 9 months ago, I would have said, correct. I'm going to wait. I'm going to wait until 3, 4, 5, 6, 7 months before I try the medicine, because I don't know if it's going to work, and we have all these other things to try first. And you know, I just don't think that's not the way we were trained to think. Okay. Now, regardless of the severity of the patient, and perhaps the more severe, the more what I'm about to say would apply, I'm ready to jump in earlier and start it sooner. It's a little bit like MCAS, and I think this is a conversation Tania and I have had. In the MCAS world, the way I initially learned it was you start with in quotes, tier 1, meaning histamine 1 receptor blockers and histamine 2. So Zyrtec and Pepcid. That doesn't work, then you add maybe a plant flavonoid. And if that doesn't work, then you add a mast cell stabilizer. And if that doesn't work, you add a imatinib or something like that. The question that I ask is, why am I putting the patient through all of that? Yes, there's a risk benefit ratio, computation to make with the drugs. But if I have good reason to think getting them on cromolyn first is going to help faster, why am I spending the time the other way? And the same argument applies to the use of this drug. I think I think it should be considered sooner rather than later, is how I would answer this. Tania, what do you?
Yeah, no, exactly what we said earlier. I mean, I think that I definitely put this up higher. Listen, but there are patients where this is not going to be appropriate
Yes.
for, and I do think you have to have conversations, with patients about it and understand, I understand that I certainly have patients with motility issues, for instance, who actually do quite well on GLP-1s. But if I have a patient who's vomiting every morning due to their gastroparesis or whatever else is going on, this may not be the first thing I try because I know that they may vomit more potentially in the beginning. I don't know 100%. It may have the opposite effect, but again, I'm going to screen my patients. I'm going to understand whether it's worth again, potential risk. It's always about informed consent. But yes, I do think that we can intervene sooner with these drugs than we thought. Um, but again, in very individual to individual.
So to maybe summarize the discussion here, what would you say to clinicians who are eager to treat their patients, but they really don't know where to start? And maybe they have a little bit of experience with other therapies, but not to the extent that you have. How can they be comfortable in utilizing this new intervention and seeing how their patients react?
I think there are actually a lot of resources online, lots of podcasts and YouTubes and discussions from individual physicians. I know I have one podcast or more than one on GLP-1s. I'm sure Tania has as
I
well.
do too. Yeah.
Yeah.
And lots of social media stuff.
Yeah, lots of social media stuff and lots of publications, um, and the RECOVER discussions. So, it's not that hard to find that information. I think the challenge is to then make the next step, or the willingness or ability to take the next step. Um, you know, I, I I welcome people asking me about that. Other physicians, I'm
Mhm.
fine with that. To the degree that I can fit it into the 24 hours in each day, I want to teach and help. So, that may be another way. That's just my way when I do it. And if they talk to Tania or Stuart, it's their way. But without label indications, that's all we got. And it's there. I guess what I'm trying to say is there's actually a lot of information out there. It's very reachable and findable, and there are people who want to help. The physician has to be open-minded and willing to take that next step.
Great.
And I think, and I'll just add, that I think David's the same as I am. I mean, I think at least when I started in this world, I would have a patient, have a conversation, and, let's say was interested in trying something that I hadn't tried before. I would tell the patient, I have to be honest, I've never used this in a patient this way, or whatever, or I've never used this drug. I've spoken to my colleagues, I've read the literature, I've done my homework. Um, I think you could benefit. I think you have to be transparent with patients. And I just say, look, we've been hunting down an answer. We don't know. There's a lot of precedence for this. So let's, are you up for trying it? And the patients will say, yeah. So what do we need to do? And so then you reach out, as a physician, you can reach out to David, you can reach out to me, you can reach out. I'm putting together that training course. And maybe there are ways that we can support as many people as we can, within 24 hours of the day. But again, like, I think you just sometimes have to take a chance, and you have to, as long as the patient's aware. A lot of my patients know, that I've quote unquote experimented with them in a way, right? We've tried something that I haven't tried in other patients. And they're actually excited about that, right? They want to be the first one, and if it works, then the next patient I say, okay, I've done it in one patient. I think there's hope here. Let's keep, let's try it. I think that's all you can do, you know.
I think for it's important for other physicians to understand. I'll speak just for myself, but I'm almost sure it's true for Tania and Stuart. They need to understand we didn't know this stuff before. Okay. So if you roll back the clock for me to 2012, 13, 14, that's when I started doing this work. Okay. Before that, I was a general internist and an HIV medicine doctor. I could treat HIV disease in my sleep by that point, by that point, live through the worst, darkest, horrible days of that plague. And we didn't know anything then. All right. And it's not so dissimilar. It's not so different from the experience with ME/CFS and now with long COVID. And the but there is one huge difference, and that's the internet. And I would argue that any physician can go online, they can go to Perplexity or ChatGPT, and they can get this stuff literally in 25 seconds. You just ask Perplexity, please summarize all the different papers and publications with the use of GLP-1s in inflammatory illness, and you get it all there, and you can start leveraging that and learn and make the next steps if you're willing to step outside the box a little bit. But the takeaway for me is the information is out there. It is reachable. You can get it. You can get help, and it's worth pursuing. You will be a better doctor, and you'll feel a lot better too, because you'll watch the difference in your patients.
Yeah. Couldn't agree more.
Wonderful. All right, we have a few questions from the audience. Can tirzepatide be prescribed for patients under 18?
According to the FDA, no. I think there are some GLPs. Wait, tirzepatide might be the one that's 16.
There's one down
are
to
now,
12, I think, too.
and there's
I
one
think
down
one
to
of them is
12.
approved at 12, yeah.
Actually, it is tirzepatide, I think. I think semaglutide is 18. Yeah, they keep doing the trials and seeing that, the side effects are low. But I think, yeah, let's check, make sure. I think it is tirzepatide.
All right, another question. Dr. Dempsey's remarks about appetite are interesting.
No, it's not. It's not. I'm sorry. It's not approved yet for 12.
Okay.
But there are a few that are approved, and I just don't remember the names. But yeah. Anyway, go ahead.
This particular person was interested in 16. So if it is approved for 16, that would answer.
It would be
question.
off-label. It would be off-label.
Yeah, it'd be off-label. I mean, it just means they didn't do the trial with kids under 16. It doesn't mean it can't be used.
Right.
It appears you can use liraglutide, which is also known as Saxenda,
That's
and
the one.
you can do for weight management greater than 12 years old and diabetes. And then semaglutide, which has many names, including Wegovy, et cetera, you can use for obesity greater than 12 years of age. But I think your
Okay.
BMI has to be greater than 95. So this is all, it would be all off label for long COVID.
Yeah.
I do not treat children. As someone who has a child, I've been given that grace to not be able to treat children, but my heart goes out to parents who have children with long COVID.
Yeah.
So the next question says, my family member has taken about 4 microdoses of Zepbound once a week. She has noticed an extreme increase in appetite. Is also on metformin for PCOS. Might the combination of the two be affecting blood sugar levels? She has not noticed much difference in her extreme disabling POTS yet, and her ME/CFS is severe.
So
But
she
yes,
had
Yeah.
increase in appetite. Is that what you said?
Yeah, the GIP portion of tirzepatide can increase appetite a little bit. And so that's why people have less side effects, theoretically, combining the GLP, which is very appetite suppressing, with the GIP, which is not as appetite suppressing. So that could be one thing. It's just that she's reacting to it paradoxically a little bit, or just idiosyncratically, I should say. It could be certainly like a hypoglycemia type thing, which usually these drugs do the opposite over time. They cause less, reactive hypoglycemia, but the drugs work by causing the beta cells to release more insulin. And if she's not eating enough, and a lot of these patients, again, like they stop eating and then all of a sudden realize that they feel awful and then they have to start eating. So there could be because of the insulin response, could be a combination. I think everyone, again, this is where it gets to, like, everyone is different and everyone's going to react differently. And some patients, POTS gets a little worse. Heart rate can go up with these drugs, both semaglutide and tirzepatide. For some patients over time, the POTS gets better, right? This is what we're saying. Everybody's different.
I think one other possible explanation for this particular example. So our patients, one of the ways that patients describe themselves and in medicine or physicians do is this idea of a sick, S-I-C-K, sick syndrome. I feel sick. And, there's lots of descriptions in the literature of what that means in terms of vagal and brain and everything else. But at the end of the day, when you feel sick, you're not very hungry. And I think that by reducing inflammation and improving this overall milieu for the patient, their terrain, that may be a reason their appetite recovers as opposed to hypoglycemia. I don't have proof of that, but that's just a thought.
Yeah, I think that's reasonable. Mhm.
My added thing would using a continuous glucose monitor. You can buy them over the many
Yeah.
startups kind of are like in this field. Low glucoses. So if you get a flag of a low glucose, they're not so good at the low glucose area. So doing a finger stick one that you could just buy at a CVS or Walgreens or whatever would help to confirm if there's actually some hypoglycemia going on. I do have a hard stop. I actually have to go see a patient right now, but I was really enjoyable to talk with all of you and nice to see everyone again.
Thanks, Stuart.
Thank you,
Take care.
Dr. Malcolm. Take care. All right, so our next question is towards subtyping development. What would each of you suspect might be needed in order to have a better chance of identifying predictors of positive or negative responses? How would you get closer to consolidating what we know thus far?
So the question is, can we predict are there ways we might be able to predict in advance who will be a responder?
No,
I don't think so. I mean, we would
no,
never have predicted this response in the first place.
that's right.
Keep in mind, we're having patients respond at 1/10 of the starting dose of these drugs. So I don't know how we could predict it. I would reverse it and say, why not give it a try in a patient when you explain the whole reason to the patient in an informed consent and you think it through, but why not? What's the downside versus the upside? Which again is what medicine is all about. It's all about risk-benefit decision making. And to me, the risk is small and the benefit is huge, so I would give it a try, even though I can't predict in advance, you know?
Yeah, I'm okay for another like 5 minutes, but I wanted to make a point because I'm gonna have to leave soon. That one thing we didn't talk about, but I see some questions about this, is the question of like why is this why is this working? Why is it reducing inflammation? And again, the paper that I was a co-author on looked at it from a mast cell perspective. Now there may be other mechanisms, but I think this is really important to understand is that and this is different from this question, but I just wanted to make sure that to cover this, the mast cells have GLP-1 receptors, GIP receptors, glucagon receptors, but lots of macrophages, other white blood cells, lymphocytes, the heart, the GI tract, you name it. There are GLP-1 receptors and most likely GIP receptors. And so these drugs are binding to the receptors. In the case of mast cells, binding the receptors, stabilizing the receptors, reducing the release of inflammatory cytokines and inflammatory mediators. And that, I think, is the mechanism that we're seeing in a lot of our patients. Again, there may be more, but that's the lens that I look at things through. And so I know we didn't really talk about the why behind it, but that's what I would say.
And if you think about that, as you decrease the inflammation in that local tissue unit, you are decreasing inflammation around the capillaries. And so in the process, you may be increasing blood flow or at least getting red blood cells through those capillaries. You get oxygen in, and then you start the reverse of the inflammatory feedback loop.
Makes sense. Exactly.
I mean, the fact that the receptors, the GLP and GIP receptors, are everywhere in our body at this point tells us something from an evolutionary point of view about the importance of those receptors and therefore the potential importance of stimulating or responding to those, agonists, drugs for those receptors. And so it begins to make sense why what was originally done for weight loss actually has this widely systemic benefit, including neurologic benefit, and there's some evidence that most of these drugs don't even cross the blood-brain barrier, but they still work. That brain fog is one of the first things that patients will tell you improves. So it may be what you're saying, that it's improving because of changes in circulation and nutrient and delivery and removal, et cetera.
Veronica, you had a question or comment?
Thank you. Yes, a couple of comments, and I'll make them very quick. I think this that you were talking about, the important role of the immune system on this, it makes total sense. And I think one of the many ways in which pacing is helping patients is because the same thing. I mean, the more energy you can allow to the immune system, the more you're able to function. Because after, probably because I'm an immunologist, but coming from the immunology side, basically we are alive thanks to the immune system. So if we keep it busy with things that are not keeping us alive, then the more we get this balance. I mean, it makes total sense. And on the other hand, what you were suggesting about, this medical doctor training that is so necessary, I completely agree with what one of the previous doctors was saying about, I think medical doctors are very afraid of taking this step because it's so many topics that they would have to tackle. I think it is overwhelming. Like many times we are saying, wait, why don't they do it? But when we start going deeply into everything that relates to long COVID, there is so many topics to tackle, and it's very hard to pick which one you're going to start with because there's so many layers to each single patient. So I think people without training on this would really make it hard. And I really appreciate the fact that you are so open to train other doctors, because I've been willing for a while, and I've been talking to Isabel about this, to do a model of train the trainer, because I think that's the only way it's going to work. We need small teams of medical doctors who are willing to be trained, and then they can expand the knowledge that you guys, have and that you want to share, because otherwise it's very, very hard. And I just would love to propose that if we could set up some type of model of this way, that could help as well to have those publications on kind of case-based publications, because it will multiply as well what your guidelines, if we can call them kind of guidelines, would be. So that would be it. I mean, of course, I have 1,000 questions, but more than anything, I'm very, very thankful to both of you for everything you're doing.
Thank you.
Okay.
Thank you.
Um,
Great ideas.
There's a question about the standard pen. What about counting clicks to microdose from a regular amount of pen where compounding is not available?
That
The
definitely
only,
can be done, right?
but I think right now semaglutide is the only one that has the clicks. Mounjaro, at least in the United States, doesn't. My understanding is there are some countries that have Mounjaro that have clicks. I want to say maybe Australia might have the
Yeah,
pen
Australia,
that does that.
Australia is one of them. Yeah.
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 And then you have the vial that of tirzepatide that you can get from Lilly, but my understanding of it is that because there is no preservative or anything, I'm not clear that you can microdose from one vial, but I don't think you can keep using that vial. That's
You're
my
not supposed to.
understanding, but you're not
Yeah.
supposed to. Right.
I mean, the click pen is the device of choice because you can then
Yeah.
rigidly and carefully and precisely control the dosing. Unfortunately, as Tania said, it's just this one product that has it. I don't know why some enterprising compounding pharmacy has not put this stuff into a click pen, because for me, the biggest challenge of prescribing this from compounding is the confusion between milliliters, milligrams, and units. And
Yes.
and there's dosing error, which is
Good.
concerns me. It's scary.
Yeah.
And the pen would solve the problem in a second.
Yeah.
So anyone want to do a compounding pen?
How are we doing with time for both of you?
Yeah, I probably have to, it may be one more question. And then I have to try. How are you doing,
And how
David?
about you, Doctor?
Yeah, I'm okay. I'm all right. I actually thought it was
Okay.
longer, so I blocked 2 hours.
Okay, great. So the next question is, does the tirzepatide also help with food noise and cravings, like with Ozempic? I love Ozempic. I only started it out of spite to show a weight-obsessed doctor it wouldn't help me. Joke was on me, haha. Super slow dosed it for 8 to 9 months to avoid side effects. Inflammation was better, pain was better. My gastroparesis is better, not cured. Cognitive is functioning less though. But that might be because I am doing more, and that's the question.
So what's the question? The question is
So food
tirzepatide
noise, is that what the
food
question
noise?
is?
Oh yeah.
It-
Yeah.
And if you
Yeah,
think the
same way.
cognitive functioning being less is just because of doing more, not because of the
They're
medication.
feeling good and going out of the energy bound.
Yeah, it's possible, but not sure. I would have thought that that going outside of the envelope would cause more fatigue and PEM than worse cognitive, but they're often very paired, they're associated.
I have seen some patients who do feel a little worse in that regard. And I don't know what to attribute it to, other than it's like, again, maybe very specific to them and their reaction to the drug. And that's where you have to. So this is a patient who's on semaglutide, or not patient. Is this a provider on semaglutide who wants to know if tirzepatide has the same effect? Is that the question?
No, the question was just in regards to the the cognitive functioning being less, and whether that's just due to doing more, or I guess if you're seeing
Okay.
that. Yeah.
But this
Yeah.
is someone who's on semaglutide. That's
Yes.
what I'm asking, right?
Yes.
So I would say that it may be worth trying the tirzepatide
Switch, exactly.
to see if it's a different effect. Yeah,
Keep in mind that tirzepatide has another receptor agonist. It's not a
that's
single,
right.
a mono
That's right.
agonist. It's a dual. And we don't fully understand the role that the GIP agonist is playing, except that it's clear, well, it's clear to us, not in a trial yet, but clear to us that it reduces the side effects, but it may also have other unrelated to side effects benefits. And for example, maybe the GIP receptors are more active in the brain. And so, using that agonist would help. I just don't know, but that's a
Right.
perfect example of when you might want to switch that you asked earlier, Isabel.
Exactly.
Someone had a question about cost, if you know, for the compounding pharmacies that you use. What are the patients?
They're all different. They're all
Okay.
different. Yeah, I think we can't answer that.
Any ballparks?
I couldn't even remember off the top of my head.
Yeah, ballpark. No, the ballpark is in, maybe in the range with shipping of $200 for a month of
Per
drug.
month? Yeah, that's probably about right.
Great. Do you know if the RECOVER TLC clinical trial will use microdose GLP-1 or standard dosage?
I'm not sure you want to get me started on this.
I think we might.
All right. Well, I hope I don't get in trouble then. So I'm on that planning group. And as you probably can guess, I am pushing very hard for microdoses to start. And don't forget, this is a government trial. It's an NIH trial. And the goal of the trial, one of the goals is for FDA indication for long COVID. And the general direction is they have to follow the rules and start with in tirzepatide, start with the 2.5 milligram dose. I could not disagree more strongly. And my fear, and frankly, the Scripps trial, which is the other trial that's cooking, which is 1,000 patients, so it's a fantastic trial, 1,000 patients in RCT, they are using the starting dose 2.5 milligrams. So my fear is that both these trials will end up failing because of patient withdrawals and side effects, and they will actually destroy the usefulness of these two drugs and force people like us to be off-label constantly and now fight all this bad press because patients will hear, oh, the drug failed in the trial and you're going to give me this drug? You're crazy. So I'm obviously feel strongly about it, but that's where it's headed. And I don't think I can head it off either. What were you gonna say, John?
I was just going to say there's a reality in these trials of the economics. If there are 20 million people that have long COVID, and if they could be helped with a GLP-1, a tenth of the dose, presumably at least it's a quarter of the cost or something, makes a huge difference.
Yeah.
And I don't think we're talking about that as much in these trials. And it's very real. Hyperbaric oxygen is work, it gets oxygen in, but at how much cost per person, time and money. Anyway,
Yeah, no, I
different
agree.
topic, but really scaling this as a societal thing is a big deal.
Yeah.
All right. So another question is, how are you deciding which drug to start with a particular patient?
I mean, I'm always
Okay.
using
Yeah.
tirzepatide first because that's the one I've had the most experience with, and it seems to have the least side effects for the reasons we've discussed. So yeah, for me, there's been no decision. I just start with tirzepatide.
Same for you, Dr. Dempsey?
Yeah.
That doesn't mean we're right, okay? Because again,
Yeah.
there's no trial. There's no head-to-head trial. You'll never see a head-to-head trial because company A is never going to compare its drug to company B. Back to the economics. So we won't know. And one of the arguments in the Scripps and the TLC trial, about starting at 2.5 has been from other doctors or people in the planning groups. Well, maybe 2.5 will work. We don't really know. And they're right. We don't know that maybe starting at 2.5 would be fine. My concern is if it's not fine, the trial is going to be destructive.
Right.
And we know that starting low does work with less side effects.
Right.
So
maybe the drug companies like Mounjaro, I mean, like Lilly, or, have to create a lower dosage.
right. Right now, they're the
That's
ones
commercially
pushing for
available.
but they're the ones pushing for using the starting dose.
I know. I'm sure.
They I know that Eli Lilly is doing a trial of microdosing, I believe is in ADHD. I can send you the information where I
Wow.
heard
Yeah, I
it.
didn't know that. That would be very
Wow.
interesting.
Yeah,
That's
that was
interesting. Yeah.
yeah, it's very recent. I listened to a podcast with their CEO, and that's what he was
Interesting.
discussing. Yeah. Okay, next question
Okay,
regarding
I'm so
reactive.
sorry.
No
I'm sorry,
Yes,
I have to leave.
absolutely. Yeah, thank you so much for being
Thank
with us
you.
today. All
Thank you. It was
right,
my pleasure. All right.
take
Take
care.
care, everyone.
Bye.
Bye.
Bye, Tania.
Bye.
Take care.
Bye.
So regarding reactivated viruses, are you following the work using Mensa, media enriched with newly synthesized antibodies to get an immune snapshot of host responses in long COVID?
I don't know anything about that. And
Okay.
I'd love to hear about it. Whoever asked the question, if they could send a link. I'm not familiar with what they're describing.
Yeah, that would be great. If you can send us, where would you like it to send? I can have them email it to me and forward, or if you'd like
Yeah,
it
why don't they send it to you and you send it to me, or put it in the chat
Put it
board
in
in
the
the
there.
questions.
question
Yeah.
answer. Yeah.
Okay, perfect.
Excuse me.
Okay, so the next question is, has your approach changed or the response of patients where a primary driver of inflammation is persistent misalignment of the craniocervical junction? So for people who have mechanical issues.
Has my perspective changed? Is that what you said?
Has your approach changed for prescribing the GIP, GLP treatments if the primary driver of inflammation is
Oh, I see the question.
mechanical
So
issues?
to rephrase it, I think the question is, if a patient has cranial cervical instability that is clinically significant, would I use a GLP-1 first? Is that kind of the question? The
I
answer
think
to me,
so.
the answer to me is no. I don't think we have drugs that can fix structural abnormalities and anatomical problems. And it's, if somebody has tethered cord or May-Thurner syndrome or craniocervical instability with brainstem compression, I don't believe that we can fix anatomical pathology with a drug. That's like a subgroup of our patients, and it's a very difficult and frustrating subgroup because none of us wants to recommend surgery because surgery has all the complications and risks for all of us, and even more for our patient population. But if you have brainstem compression, it's got to be fixed, and I don't think a drug will do that. I think a drug may help before and after, but it's not going to fix the anatomy.
Yeah. Okay. Have you had patients who were underweight who benefited from a low dose GLP-1?
Yeah, most of my patients are probably underweight. They're either at a reasonable weight or underweight, and they benefit. Again, we're using a tenth of the dose, so we don't want them. I almost never want them to lose weight. I mean, one of the problems is I want them to have more muscle mass, which implies the opposite of losing weight, and that's one of the risks of weight loss dosing of GLP-1s, the patients lose muscle mass. So I don't want that to happen. And I will watch that carefully and often suggest, some creatine or protein type supplements to try to maintain muscle mass. But I haven't seen it as a problem.
We have a 33-year-old patient who has been bedbound for 3 years that asks, how do you reduce inflammation systemically? And I'm assuming the question is besides the GLP-1.
Well, GLP-1, but also, I mean, I guess the first question I would ask is what's going on with the patient? I mean, that's not enough information, but leave it at that. So the patient's bedbound, I presume over the 3 years, there's been some evaluation and workup. Rapamycin can similarly reduce inflammation. GLP-1s can, but I would suggest that we're getting off topic, that any patient who's bedbound for 3 years needs a careful anatomical workup for CCI, tethered cord, and things like that. My experience has been a significant percentage of bedbound patients end up having CCI, and that's why they're bedbound.
Mm.
That's a pretty broad statement, so I probably should be more cautious, but.
Mhm.
Just a quick note on the autophagy and rapamycin and mTOR. The Thomas Bunker did a long COVID study with restricted eating that was just in Nature pretty recently that had good effect of changing some of the autophagy and symptoms at 4 weeks. And there's when you time restrict eating or fast, you end up changing something called AMP-K, and AMP-K is upstream of mTOR, and so it turns down the same thing that rapamycin is turning it down, but does it indirectly. And that also is how metformin works. And metformin will have AMP-K and also berberine. They'll have AMP-K effects at lower doses in general too, than as compared to the diabetic doses that are trying to keep new sugar from getting made out of the liver.
Right. What proportion of your patients respond positively to your treatments? Not just these particular therapies, but generally in your practice?
Well, I mean, it's a very broad question because it's hard to, you know, are we talking about a little response, a medium response, a great response? I would say managing POTS and mast cell and motility disorders and leaky gut, if those are managed well, a very significant portion of patients improve. They're not cured, they improve. You know, it's often if going up is getting better, the pattern I often see is a sort of an initial relatively steep trajectory as we add meds for these problems, is what I'll call the low-hanging fruit, and then they start to plateau. I think that interventions like tirzepatide and rapamycin and other things that we haven't gone into can speed up that trajectory and hopefully avoid that plateau and get them back to a real state of health. So I don't want to make it sound like I'm some genius, but yeah, a reasonable proportion of patients respond as we titrate and trial and error all these drugs for these drivers of their illness, which are mostly the perfusion issues from POTS, leaky gut, inflammation, and mast cell activation.
So in your sensitive population, do you have anyone who can't tolerate the injections themselves?
Tolerate in terms of side effects, you mean, or doing, well, some patients are afraid of needles, so that's definitely an issue. It's a small group, but it's real. And I'm starting to try oral GLP-1 for that. I've not had anyone not tolerate the injections, even though they're often mast cell patients. And again, in this very low dose, I had one patient this week, actually one patient this week who said that she took 1.25 milligram dose and crashed. And I don't understand what happened, and we're trying to figure it out. But that's literally the first patient when that's happened.
Okay, so we have the last question, and then if any of the panelists have any additional questions. I'm a 3-year bedbound patient. who could not tolerate metformin or Victoza. How about rapamycin? Is the medication rapamycin? And that's where the question ends. So maybe the question is if you think that would be better tolerated.
I think it may be the patient who talked about being 3 years bedbound ME before and is trying to get clarification. Is the medicine that we were speaking about rapamycin? And it is rapamycin.
Right.
And low dose rapamycin, and let's emphasize bolded, italicized, and underlined, low dose, has very few side effects. It's very, very well tolerated. If you tell your physician, your PCP physician, oh, I want to try rapamycin, they'll think you're crazy because they will appropriately associate it with immune suppression for a kidney transplant. At this 6 milligram dose, it's like so many other drugs, this hormesis, where it has different properties at different doses. At the low dose, it really is well tolerated. I mean, we're doing this trial, and you know, occasionally you get mouth sores, but I've not seen increases in infection or any of the other side effects that people worry about.
Thank you. Does anyone else have any other questions?
Yes, I was wondering, are you trying rapamycin and the GLP-1
Yes.
agonist?
Yeah, I have been using them both. I
Okay.
don't start them together at the same time, but I will add one or the other depending which the patient is on. And I see additional benefit, and I've not seen increased side effects.
Okay. And have you seen any effects on PEM?
PEM is the $1 million challenge, you know. I think most patients who are responding have said they have more stamina, a bigger envelope. If they get PEM, they recover faster. So they have less PEM and they recover faster, but it's still something we have to attend to and watch out for. And I think, as somebody said earlier, you know, one of the classic problems in taking care of our population of patients is when they feel better, just like if I was sick and felt better, I want to catch up and take care of everything, and then they overdo it and they pay a price. And it's very frustrating, and that's something we always have to keep talking about together.
Yeah, of course. No, because I was kind of following this line of thought that John put out earlier about, you know, increasing this blood flow, because I've always wondered how much of the PEM is coming out from the microcluts.
Yeah.
And how much is coming out from, of course, other things that we still do not know. So this will make sense. But of course, as you know, well, as you were saying, it has to be kind of a slowly adjustment of the pacing more than anything. Okay.
Right.
And you
And
can
I don't.
certainly.
Go ahead, John. I'm sorry.
Oh, I was just saying, and you could certainly hypothesize that, you know, if PEM ultimately is some kind of a metabolic recovery, or in somebody who's already in a glycolytic state, and there are a bunch of out there kind of guesses along that way. But if you're sitting at the edge of your energy availability, like somebody who's run a marathon, and so you're not using your aerobic metabolism, your TCA cycle, then if you get that blood flowing back and can get more oxygen in and more of the materials and get HIF1 out of the nucleus and NRF2 into the nucleus and so on, it theoretically should help to balance the available energy. But that series of statements is very different than, can you take a magic wand and change PEM, because it's not that easy.
Right. Just to emphasize what you're saying, John, I don't want to sound, I probably have sounded, and I don't want to sound like tirzepatide is the cure. Okay. This is a phenomenally complex illness, and we're not going to find a single drug or a single intervention that cures it. I think it's going to be a, you know, constant trial and error combination of things. And if you look at the hypotheses of what drives long COVID now, as of today, it is so complicated, and we've only just started. I mean, we move remarkably fast if you think about it, but as John points out, we're talking about all these metabolic pathways that interact with each other, and the outcome is this phenotype long COVID. But it's very complicated when it's not going to be a single intervention.
Yeah, no, I completely agree with that. But I think this kind of findings can help us to prioritize where to
Yes.
start, because I think that's one of the main problems. I mean, I'm not a medical doctor, but I could see it when you start reading about everything that needs to be done. And I completely agree, this has to be a multi-systemic and a multi-drug approach and a multi-other tools approach. But that's one of the main problems. Where do we start from with each patient? Because, it's very hard. So if we could find this kind of pattern of what could help in steps, I think that could make a huge difference.
And
Yep.
if you go to the literature on the hypoxic theory of disease, people have been publishing about that for a long time. I thought it was really cool that, oh, hey, this would all make sense from a plumbing perspective. And then, like most things, somebody figured it out a long time ago. But it's anchoring on what that means and how to reverse it may be helpful to some.
Dr. Kaufman, what's the lowest dose that you would start at? And are there any specific tests that you would want to see before starting tirzepatide?
So I usually, excuse me, I usually start with 0.25 milligrams. With 1 or 2 patients, I've tried to start lower. It gets very difficult because of the insulin syringe and the volume of the drug. In terms of labs, I mean, when I see a new patient, I do a pretty large lab order. My patients, if anyone knows my patients, they will tell you the phlebotomist does not like my orders because it's 30+ tubes. So I already have a ton of information. I think at a minimum, you'd want to have a CBC and a complete metabolic panel, thyroid tests, probably amylase and lipase to check pancreatic function, or not function, but condition. Those would be the main things, I think. Yeah.
And the same question in regards to rapamycin.
Okay, so rapamycin's a little different. CBC, complete metabolic panel, but there I will check a lipid panel and a hemoglobin A1C, because rapamycin can increase cholesterol and or glucose intolerance or insulin resistance. That's probably dose related, so I actually use that to help titrate the dose down if I see that. I usually will get a high sensitive CRP to follow inflammation, because that's one way I can show that it's reducing inflammation. And then I monitor those labs monthly, at least
And
for
the
the first
starting dose?
at least for the first 3 months. So I tapered up. The target dose is 6 milligrams a week, once a week. And I used to go 6 weeks to get there. Now I go 3 weeks because it's been so easy. And frankly, we probably could start at 6 milligrams, but I have not gotten that courageous yet.
Yeah.
But 6 is the target, not, we don't know the right dose. That's the target dose based on clinical trials in people, not mice. And then you adjust from there, up or down.
And then in terms of the low dose GLP-1s, and you may have said it at the beginning, but if the starting dose is 0.25 and it's a tenth of the starting dose of GLP-1s in general, is there a target that you typically try to end up with on low dose or microdose?
So again, I hate to keep saying it like a broken record, but it's trial and error. I will keep going up. Although I mostly stop at 1.25 because I don't want weight loss, and I worry about their dysautonomia and gut motility. However, I have patients on higher doses because they want to lose weight and they're okay. So maybe I'd get better results going up to 2.5. I'm just not sure.
But in general, that kind of 10x range.
Yeah, exactly.
Great. One last question before we go. Emily?
Oh, I just wanted to ask if you had any patient experiences who have the opposite of gastroparesis, who have rapid emptying disorder caused by dysautonomias, who still have some mast cell inflammation, might have some mechanical, weight gain issues, a mix of these things. I know we've had some patient concerns and questions, who've been trying out these medications low doses, some of the lower regular doses, and considering there are just as many people who have to deal with rapid emptying concerns, metabolic enzyme concerns, mitochondrial disease, malabsorption concerns. I'm just kind of wondering what are some of the experiences you've seen regarding these things as well?
Well, rapid emptying probably would benefit since the GLP-1 slow motility. I'm not sure I would use it for that reason, but that would be a unexpected or taking advantage of a side effect. So that would, be a good thing. I think that I have only had patients with motility disorder, the opposite of rapid emptying, but wouldn't see that as a contraindication to using it. You mentioned mitochondrial function. So, for me, that circles back to the systemic inflammation and reducing it. And that ultimately helps mitochondrial function in the same way rapamycin does. So, these are, again, it's multiple interventions trying to attack the problem, both what we know about the problem and frankly, what we don't know, so as I'm sure John knows and talks about, there's, in terms of mitochondria, you can look at the GLP-1s, the rapamycin, red lights, metformin, acarbose, SGLT2s. I mean, they all have a role, and it's for me, the analogy is way back to my HIV days when it wasn't until we used 3 drugs at once that we could stop this virus from working. And I think it's the same approach in tuberculosis. It takes more than one intervention.
Yeah, cancer changed dramatically in the
cancer,
60s
right.
to 70s when they went from serial disease or drugs.
Exactly.
And just a quick anecdote on the red light. My understanding is that one of the ways that red light may work is by its activity on the sodium-potassium pump, which becomes really interesting in terms of this whole energy discussion, because the sodium-potassium pump is so critical in keeping the sort of electrical separation in the cells the way it needs to be for the energy production to occur.
Right.
All right, we are at the end. So thank you, everybody. And thank you especially to Dr. Kaufman and Dr. Dempsey, Dr. Hutton and Dr. Malcolm. Thank you for the work you do and for your willingness to share your knowledge with other clinicians and with the
trying to pay
public.
attention.
So, so others can benefit and we can, be able to have more choices for patients, and it's really needed. So we are all very, very thankful for the time and the knowledge that you share with all of us.
Thanks for having me on. I really enjoyed it. Thank you.
Thank you. Have a great day, everybody. Have a great weekend.
Okay. Bye.
Bye.